Hello, my name is Kira Miller, and I'm a second-year master's student in the Integrative Biology program at San Francisco State University. Imagine you're out hiking in the mountains when you walk up to a lake and see this. This mass die-off is the result of chytridiomycosis, a cutaneous amphibian disease caused by the aquatic fungal pathogen Batrachochytrium dendrobatitis, or BD. The emergence of BD has resulted in amphibian declines worldwide. However, susceptibility to BD infection varies between amphibian species. In California's Sierra Nevada mountains, the endangered mountain yellow-legged frog, Rana sierri, is highly susceptible to BD. But in contrast, the co-occurring Pacific chorus frog, Pseudacorus regilla, is more resistant and has been proposed as a BD reservoir. Although chorus frogs are one of the most abundant and widespread amphibians in western North America, this species and its interactions with BD are understudied. Amphibian skin also hosts communities of symbiotic bacteria, known as the skin microbiome. Recent research indicates that for some amphibians, the skin microbiome may facilitate survival against BD by producing antifungal compounds. My research investigates and compares BD infection and skin microbiome communities between Rana sierri and Pseudacorus regilla and seeks to better understand the extent to which chorus frogs may be acting as a BD reservoir in this system. During the summer of 2023, I visited 24 lake sites across the Sierra Nevada mountain range to capture and collect skin swabs from frogs. DNA extracted from these swabs was used to quantify BD infection intensity using qPCR, and next-generation sequencing was performed to sequence bacterial DNA that was present on frog skin. Our results show us that BD infection prevalence is high in chorus frogs, and that BD is widely distributed throughout their Sierra Nevada range. However, BD infection intensities, shown here on a logarithmic scale, were moderate in both species and well below infection levels associated with BD-induced mortality. Previous lab studies have shown that chorus frogs can carry high levels of infection without showing symptoms of chytridiomycosis, supporting their role as a BD reservoir. However, results from this study suggest that natural infections in this species may not be so severe, and the extent to which they act as a BD reservoir may depend on how much co-occurrence they exhibit with other species. Skin microbiome analysis is still ongoing. These analyses will explore and compare the relationships between bacterial composition and BD load to identify differences or similarities between a susceptible and reservoir host that exist in the same environment. Understanding the many factors that influence BD dynamics among co-occurring amphibians is important and can help inform conservation strategies for disease-susceptible species in the future.